Hi guys and welcome to my channel. Myself Dr. Akshay, and in today's session I'm going to be discussing about the topic long-term blood flow regulation. If you guys want to learn more about the circulatory system, then click subscribe and the bell icon. So let's do this together. Coming to the topic long-term blood flow regulation. Long-term blood flow regulation is especially important when the metabolic demands of a tissue change. Thus, if a tissue becomes chronically overactive, therefore that particular tissue needs more amounts of oxygen and other nutrients. So, the arterioles and capillaries both increase in its size and numbers within weeks to match with the needs of the tissue. And this process does not occur if that particular circulatory system is pathological or if it is too old to respond. Next, coming to how blood flow regulation occurs by changing in its tissue vascularity. So, a key mechanism for long-term blood flow regulation is to change the vascularity in tissues. If the metabolism in a tissue is increased for a prolonged period, this vascularity towards that tissue will also increase. And this process is known as angiogenesis. On the other hand, if the metabolism in a tissue decreases, the vascularity towards that tissue will also decrease. The reconstruction of tissue vasculature occurs in young adults. Rapid tissue vasculature occurs in new growth tissues, such as in cancerous tissues and scar tissues. But this reconstruction of tissue vasculature occurs very slowly in old, well-established tissues. Next, coming to what is the role of oxygen in long-term blood flow regulation. Oxygen is not only important for acute blood flow control, but it's also important for long-term control of blood flow. For example, we are putting premature human babies into oxygen tents. This is done in case to prevent neonatal hypoxia. Since in preterm infants, this retinal part is under development, and since this retinal part is having high metabolic rate with thin vascular layer of tissues, this retinal part of this preterm infants will absorb maximum oxygen concentration. So, we are putting preterm baby or preterm infant into this oxygen tent. And after some time, we can observe that this retinal blood vessel stops growing. And further, we can observe that this regression of retinal vessels occurs. So imagine when this preterm infant is taken out of that oxygen tent. So when this preterm baby is taken out of that oxygen tent, an explosive overgrowth of new vessels occurs. So here, if so much overgrowth occurs, that is, the retinal vessels protrude out from the retinal layer, and when it enters into the vitreous humor, this will eventually lead to blindness. And this condition is known as vitreolental fibroplasia. So with this example, we got one idea. That is, when that preterm baby is supplied with sufficient oxygen, these vascular growth factors are not produced. On the other hand, when that particular preterm baby is exposed outside, that is, when that preterm baby is taken out from that oxygen tent, there occurred a deficiency of oxygen. When deficiency of oxygen occurs, these vascular growth factors are stimulated, and this vascular growth factor made that vessels to grow in numbers. So what happened? Blood vessels increased in number and size. So when that increase in number and size raised it to more than its limit, it led to blindness. So what is the importance of vascular growth factors in the formation of new blood vessels? Vascular growth factors or angiogenic growth factors are formed as a result of deficiency of oxygen or other nutrients or deficiency of both. So there are four angiogenic growth factors, and the first one is vascular endothelial growth factor. Second one is fibroblastic growth factor. Third one is platelet derived growth factor. And finally the fourth one is angiogenic. And certain other substances like steroid hormones can cause dissolution of vascular cells and disappearance of blood vessels. And peptide hormones which are produced in the tissues can block the formation of new blood vessels. So one such example for a peptide is angiostatin. Angiostatin is a fragment of protein plasminogen, and this is a naturally occurring inhibitor of angiogenesis. Endostatin is another anti-angiogenic peptide, and this endostatin is derived from collagen 17. Anti-angiogenic substances help in preventing the blood flow towards cancerous tissues so that nutrients and oxygen cannot enter cancerous tissues and thereby depriving its further growth. So some of those anti-angiogenic substances that can be given externally are thalidomide and aflibercept. So that covers the topic long-term blood flow regulation. I hope you guys learned about this particular topic. Thank you so much guys. See you guys in the next session.